We had mentioned about the translation and that there should be a modification in the protein molecules. We should be talking about some more details about these processes so that the evolutionists will understand there may be no coincidence involved in this, because a factory setting is required, a great intelligence is required to carry out all these steps. We talked about translation and also that there should be modification of the protein molecules. A signal peptide is required to give this information, and when the protein synthesis is carried out, that signal process has a certain message, and that message is carried out by another protein molecule, and that message is placed on the protein molecule saying that it should be carried out to a certain location and a certain modification should be processed. This way they travel to the endoplasmic reticulum with this signal, and we will be watching the details in the video. When they come to the endoplasmic reticulum, we can see that they have come to the endoplasmic reticulum. We can see that there is a green part here, and this green part has the certain signal, and that green part carrying out a certain message will be attaching to a receptor. The signal recognition receptor. Now we are going to see how there is much detail in this process. There is a signal recognition protein we can see here, and that part on the signal gets attached to the receptor, and after the attachment takes place, we can see that the ribosome enters into the membrane of the endoplasmic reticulum, but there is a translocon protein gate here, and it has recognized the ribosome, and the ribosome attaches to the endoplasmic reticulum's membrane, and the process will be going on. And we can see that while the process is continuous, the recognition continuous, and we can see that inside the endoplasmic reticulum, the protein molecule is entered into the endoplasmic reticulum, and every atom is recognized here, and every atom is carried out through a certain order. And after that process is completed, if we are to rewind for a certain instant, we can see that a signal peptizase protein molecule, an enzyme, is coming towards the protein molecule, and the signal peptide should be cut at the right place. And this signal peptide actually calls this certain enzyme to the environment. However, definitely there is no coincidence here, and at the right location, the protein molecule is cut so that the signal peptide leaves the protein molecule. And this will be continued by adding another information to the protein molecule. And this protein molecule informs that the protein synthesis is completed. Here, we can see that the cutting takes place, and right at the required location, the cutting is completed, and then the translated protein enters the endoplasmic reticulum. And there are some other proteins that are on the membrane of the endoplasmic reticulum, and the translocation is started by means of that special protein molecule. However, there is another stop transfer sequence required in order to carry out the process in the endoplasmic reticulum. And while the translation is continuous, again, this stopping enzyme, a peptide, is required. And there's an internal signal sequence here that is formed, and the stop transfer sequence is again functioning so that the manufacturing goes on. This is just like a sewing machine. And this way, the protein molecule is processed on the membrane of the endoplasmic reticulum. However, the structure of the protein molecule is not destroyed here, and through this mechanism, no harm is given to the protein molecule. And this is just like a flowing and continual mechanism that takes place on the endoplasmic reticulum's membrane, and the scientists have not been able to discover all the details of this process. And after those protein molecules are entering the endoplasmic reticulum's